I'm Katie Osborn, graduate student on the MPA Estuary project, and today I'm going to be talking to you about some of my thesis research. First, I'd like to acknowledge my co-authors, sponsors, and our many wonderful volunteers without whom we wouldn't have been able to complete this work. The Mad River Estuary, pictured here, is right in Humboldt State's proverbial backyard and served as the reference site for the MPA Estuary project. Frank Shaughnessy, in the next presentation, will be talking about our results from all 4 of our study systems on the map of California to the right, but today I'm going to focus just on the Mad. So we had 3 sites, which I will refer to as mouth, tributary, and upstream. Our mouth site was near the boat ramp on Mad River and experienced direct wave action, particularly as the mouth migrated during the course of our study. Our tributary station is at the outlet of Norton Creek, locally also known by the name Widow White Creek, which is one of Norton Creek's tributaries. And our upstream station at the bend of Tyee City is just a quarter mile from the Hammond Trail pedestrian bridge. We sampled June 2014 through June 2016, quarterly in the first year and monthly in the second year. We used beach seines to characterize the fish community in the Mad River, and I will be presenting results from this data first today. Next, I will be presenting our findings on the invertebrate community, which we sampled using clam guns to take benthic cores to a depth of no more than 10 centimeters. And finally, we linked our knowledge on the fish and invertebrate communities via a diet study of 2 benthic fishes, English sole and staghorn sculpin, and I'll be wrapping up my presentation today with a discussion of those findings. So first off, we have fish abundance in red as catch per seine haul for all months when flows allowed us to safely sample. And you can see in blue why we weren't able to safely sample in the months that we didn't get out there. So these were really high flows caused by El Nino conditions last winter 2015. You can see that our abundances were lowest in the winter months and highest in summer, remaining high through fall, and that our peak abundances occurred in June 2014 and July and August of 2015. Now, these increases in abundance were accompanied by increases in diversity. But when we look at diversity, we see that the species that would contribute to those large summer catches were already moving into the system in spring, with a big uptick in April 2015 and March 2016 in the species represented in catch. We also see that diversity remains pretty constant from June through October, despite huge fluctuations in catch throughout our summer and fall sampling period. And this reflects the fact that our really big summer spikes in abundance were mostly due to capturing schools of a few highly abundant species, rather than an even increase in diversity across the board. Since the Shannon Diversity Index pictured here takes both the number of species and their relative abundances into account, it's able to reflect this fact that our summer spikes in abundance were mostly due to large catches of a few highly abundant species. Now we wanted to, and don't worry, I'll walk you through this. We wanted to verify our observations statistically, and to do this we used non-metric multidimensional scaling, or NMDS, and analysis of similarity, or ANOSIM. NMDS groups samples according to their species composition without any a priori information regarding explanatory variables. Researchers can then look at those groupings to see whether they reflect known ecological gradients, such as latitude, time of year, temperature, or salinity. Here we, excuse me, here we see an NMDS plot with a really nice separation by season of sampling. So we have this lovely progression from winter through to spring, summer, and fall. Winter, which represents our least diverse, least abundant sampling events, are clumped off separate to the side, while spring and fall, our most variable sampling months, exhibit the broadest spread. And when we look at the species captured during these different seasons, we can get an idea of the seasonal fish communities in the Mad River. So our winter community was sculpin dominated, primarily by Pacific staghorn pictured here, but also by other hardy estuarine resident fishes, such as coast range and prickly sculpin and three-spined stickleback. In spring, estuaries are key to life cycle completion for many fishes, and we start to see those species moving into the estuary at this time. Those are juvenile flatfish like English sole and starry flounder that move into the estuary as juveniles to rear and then out-migrate again in fall. And it's also salmonids like steelhead that migrate through the estuary. Summer had the highest diversity and also the most marine fishes, as a lot of marine species move into the estuary during the time of highest productivity seasonally to feed. So these were silversides like top smelt and jack smelt, and coastal sculpins like cabezon and tide pool sculpin. Our fall community was again characterized by these fish that rely on estuaries for life cycle completion. Both bay pipefish and shiner surf perch gave birth in the Mad River, and we also saw a lot more coho and chinook smolts at this time. Season was the most important factor significantly describing differences in the fish community. But the second most important factor was station location. Now I'll walk you through this one too. So here we have our three stations: upstream, tributary, and mouth. And the circles describe the standard deviation for station catch. So they're not going to encompass all of the points that represent samples taken at a particular station, but they will be representative of the centroid for station catch. And here we have a really nice upstream to downstream gradient with a lot of overlap between the tributary and mouth stations, which were a short walking distance from each other, and no overlap between the upstream and the mouth stations, which were about a mile apart. Now, to remind ourselves why there might be these differences, we're going to look at what these stations actually look like. So our upstream station had seasonal rupia eelgrass beds, cobble substrate, and some riffle habitat, and was usually at 1 to 2 parts per thousand salinity. Our tributary station represents the last freshwater input before you reach the mouth, and these waters are cooler than the ocean waters, even in the summertime. This is cobble substrate and algae. At the mouth, we had direct wave action and sandy substrate. And looking at the fish caught at these three stations, at our upstream station, we found those hardy estuarine resident species, once again, your prickly, coast range, and staghorn sculpins, as well as your three-eyed stickleback. And we also got salmonids utilizing that riffle habitat, particularly steelhead. Our tributary station was really popular with the juvenile fishes seeking out that freshwater input. Copper rockfish were usually captured here. We also got chinook and coho smolts, as well as flatfish juveniles like English sole. Now, flatfish move into the estuary as juveniles to rear, so we also caught them at the mouth, just as they were starting to enter the system. And those were juvenile sand sole and English sole. And at the mouth, unsurprisingly, is where we caught most of our marine species. So gunnels like penpoint gunnel, top smelt, which I showed earlier, and our osmerids like surf smelt and night smelt. Now, here is the same data we started with for fish, but for inverts. And since this is presence-absence data instead of count data, a few adjustments had to be made. So rather than abundance in red, we have a sort of inverse abundance, which is the number of empty cores. And you can see this really spikes in our winter sampling periods, representing really low abundances, and that it drops to 0 in fall. So there was 100% cover of inverts in the benthos. In black, we have diversity as average number of species per core, and both diversity and abundance peak in the fall, a little bit after our peaks in fish diversity and abundance. Now this is likely an artifact of our sampling regime. We weren't able to sample our benthic invertebrates in July and August when we had the highest number of fish and the greatest diversity of fishes. So if we had been able to go out there, we may have found comparable abundance and diversity in July and August. Now, just like with the fish, we wanted to look at NMDS and anosim for the inverts. So we again found that season and station were the two most important factors significantly describing differences in the invertebrate community. But these two factors were reversed, and we can see this when we look at the NMDS plots. So you can still see the seasons pulling out, but it's a lot less clear. There's a lot more overlap between the seasons, and winter in particular doesn't pull out as remarkably less diverse, less abundant from the other seasons of sampling. When we look at the station location, however, we still have a really nice downstream to upstream gradient with a lot of overlap between the mouth and tributary stations and a lot less overlap between the tributary and upstream stations. Anosim showed that the most important variable for determining the invertebrate community is station location, and seasonality is secondary. To visually compare our benthic invertebrate community to what we were finding in the stomachs of our benthic fishes, we did percent composition by functional group. Here again, you can see the decreased diversity in those winter months with a lot fewer functional groups represented in those cores. We also see that polychaetes in dark red are present nearly year-round, as are amphipods in gray. But the amphipods, despite being present year-round, still decline in those winter months. This contrasted with what we saw in diet, and opening with English sole, we see that amphipods really dominate the diet regardless of what time of year the fish are feeding. Now, English sole only enter the estuaries seasonally, so they're only present part of the year, and we only captured them part of the year. But when we did capture them and look at their diet, it was amphipod dominated in all months except May 2016. And here in May, you're seeing a copepod-dominated diet. These are herpatocoid copepods. They live in the benthos, and that's the color orange here. And we found that for English sole, they prefer copepods when they're smaller, and as they get bigger, they switch to amphipods. Now with their big smiling mouths that you can see right there, size selection wasn't as much of an issue for our staghorn sculpin. So they chowed down on amphipods year-round, no copepods for them. And polychaetes, while present in the benthos year-round, were a regular but small component of staghorn diet. We dissected nearly 400 staghorn sculpin and were therefore able to do NMDS analysis on this data as well. Perhaps unsurprisingly, with the dominance of amphipods year-round in the diet, season did not significantly determine what staghorn sculpin were eating, but the location where they were captured did. So staghorn that were captured at our upstream station had a significantly different diet, favoring a lot more insect larvae and more freshwater invertebrate species than staghorn captured at our tributary and mouth stations. So, in closing, increased abundance was accompanied by increases in diversity, but was mostly due to catching large numbers of a few highly abundant species, like three-spine stickleback here and Pacific staghorn sculpin. We found that both the fish and invertebrate communities were largely determined by the time of year and the location where they were captured, but that these two factors were swapped for fishes and invertebrates, so that the fish community was mostly determined by the time of year that we were fishing, and the invertebrate community was mostly determined by the location where we were sampling. Finally, we found that the diet of our two benthic fishes were each determined primarily by one factor each. English sole size was the only factor that affected their diet, whereas for staghorn sculpin, station location was the only factor affecting their diet. Both species ate the same stuff regardless of when we were out there fishing for them. Thank you very much, and are there any questions?
We have plenty of time for questions.
Yes.
How many of these species move up and down across your stations, and to what extent could that have influenced their diet versus just staying in one place?
So, our English sole never moved all the way up to our upstream station. We only encountered an English sole at the upstream station once. So couldn't really, you can see with our staghorn that there's a lot of overlap between the tributary and mouth stations, since our English sole in any given month were typically only caught at a single station, coupled with the fact that except for one fish, they were all caught at those two downstream stations. Couldn't really look at size differences or diet differences based on that for the English sole. Staghorn did move up and down, but again, there was that confounding factor of when we found lots of staghorn downstream, we often weren't getting any upstream. So then you have sampling time confounded with location. Yeah, Joe.
I know the high flows kept you guys out of the sight sometimes. It looks like some of the high flows you caught fewer fish. But I was wondering if you took a look at all at whether flows changed the fish assemblage when you were able to get out. And also I was wondering whether sort of those low abundances during the high flows when you were able to get out, is that real, or is that an artifact of difficulty sampling in the high flows?
So good question. I did look at flow, and there is a flow meter directly on the Mad River operated by USGS. So I did have flow data for the Mad, and it was almost significant in my analysis, but not quite. And it came after every other variable in importance. The other issue for us, we're out there in the tidally influenced part of the river. And so what really impacts the water level isn't just flows, but also tide. So I tried to get out there when the tidal level would ensure a pretty even depth across months, regardless of the flow. Good question. Yes?
Did you take a look at the effect of marine mammals in the estuary?
So the only marine, I did not take a look at marine mammals in the estuary. We have river otters and seals that hauled out on the far shore. But other than
Ruining our crab traps.
Oh, well, I mean, that's crab traps though, so it didn't affect my data.
It affected my data.
Yeah, Frank can answer that question. The seals sometimes were stalking our beach, staying on the outside, but that only happened a few occasions. Any other questions?
Thank you, Katie.